All right, welcome back, guys. In this video, we're going to continue our discussion from last time about free energy and chemical reactions to add in more details about enzymes. So, quick recap. Exergonic and endergonic reactions. We're asking the question, how much energy is either released from the reaction or is needed for the reaction to happen? And remember, free energy just means potential energy that are in the bonds of that molecule. And so if your reactants start off relatively high and the products are relatively low, you've got an exergonic reaction, which means energy is going to be released. And we can harness that energy to make ATP. You can release it as heat. In reality, it's usually a combination of both. Right? In an endergonic reaction, right, our reactants start off relatively low and the products end up high, which means we had to put energy into the system. Okay, so that's why, remember, exergonic reactions are a negative delta G and endergonic reactions are a positive delta G. Right? In biology, this almost always means catabolic reactions on the exergonic side, and it means anabolic reactions on the endergonic side. So what do? We take in energy in the form of food and calories, go through catabolic reactions, right, converting to ATP along the way, and then use that ATP to fuel these anabolic reactions that require energy input. Right? Now the catch is, in order to make these reactions happen, we have to get over this activation energy hump. Right? And activation energy just means basically the amount of energy that you need to get the reaction started. So I like the analogy of a ball rolling up a hill. If you want a ball that starts here to wind up down here, it would be easy if they were connected by a straight line because the ball would just roll down the hill. But we have this activation energy hump, which means we have to start breaking some bonds in our chemicals if we're going to get the reaction to proceed. And so you can add activation energy in the form of heat. That speeds up the reaction energy because heat makes molecules move around faster. Or you can add something called a catalyst. Right? And all a catalyst does is reduce that activation energy hump so it's a bit lower, and now it's easier to push our ball over that smaller hill. And remember, the reactants and the products, they stay the same. So even if we add our catalyst, which is going to be our enzyme here in a minute, the delta G doesn't change. Right? Reactants start here, products start here, same exact molecules. We're just making the reaction happen more rapidly. Okay, and we can lower activation energy for an endergonic reaction too. You're just seeing that here. But the same principle, reactants and products don't change. And so in this case, our positive delta G is still the same. All right, so as a summary of how enzymes work, let's use this diagram. The idea here is this gray blob is supposed to represent the enzyme. This green molecule is supposed to represent a substrate. Now, the active site is just the binding site for the enzyme, and they're doing this on purpose. They're showing you that the shape of the substrate and the shape of the active site aren't exactly perfect. Right? This entire idea is called induced fit, and what it really means is when two molecules bind to each other, their shape is going to change a little bit. And so induced fit just means when the substrate binds to the active site, both molecules sort of twist and shift just a little bit to make so that their shapes fit together, almost like two puzzle pieces that click together, right? And once they do, then you produce this enzyme substrate complex, which just really means the enzyme, this guy, and the substrate are bound to each other. Once that happens, you can actually have a reaction. In this case, we're going to be splitting these two halves of the substrate molecule apart. So this would be a catabolic reaction because we're taking a large molecule and breaking it apart into two smaller ones. So temporarily, you get this state where you have the enzyme and product. So the conversions happen. It's just that the enzyme hasn't quite let go of our two product molecules yet. Eventually they do. Products are released and are free. And now your enzyme is free to go back and bind another substrate molecule and start the entire process all over again. Right? So the way that I like to think about this is when we're lowering activation energy, that really just means that this enzyme is applying some tension to that bond. It is physically pulling on the two halves of that molecule, and trying to pry them apart, that tension, right, or pulling and twisting on the molecule, that is what's actually lowering our activation energy. And in the end, all that does is make the reaction go faster. Okay? So when you see a free energy curve, this is the same concept as the ones that we just saw a few minutes ago. Remember, reactants don't change their energy level. Products don't change their energy level, right? So our change in energy is exactly the same, but our activation energy goes down. The end result is we just get a faster reaction because it's easier to overcome that activation energy hump. Okay, so when they say that the red dashed line is the reaction with the catalyst, in a biological setting, that pretty much always means with an enzyme. In chemistry, there are catalysts that aren't enzymes, but in biology, they're pretty much all enzymes, and enzymes are almost always proteins, so we can regulate those enzymes to control how fast these reactions happen. Okay, so for this graph, we're going to be talking about inhibitors, but I want to set up what the graph is even showing us in the first place. Remember that the substrate, it's often written as an S inside brackets because in chemistry, these squared off brackets always mean concentration. So substrate concentration just means how much of our reactant do we have, right? Because the substrate is what binds to the enzyme to begin with. And with just the enzyme by itself, it's pretty straightforward. This purple line, if you add more and more of the substrate, the rate of the reaction, right? You could think of this as like number of moles or molecules per second that are being reacted. If you have more substrate, the reaction goes faster. And the reason for that is enzymes and substrates don't actively seek each other out. It's not like the enzyme can go and find the substrate. They just happen to bump into each other randomly. And the more substrate molecules we have, the greater the probability is that they're going to happen to bump into each other. It's like blind people walking across or around a room. If everyone's blindfolded, the more people there are in the room, the more likely you are to have a collision. Same deal. The more substrate molecules you have, the more likely you are to have a collision with the enzyme, so it goes faster. At some point, we hit a maximum rate, and the enzyme can't go any faster. That max we call Vmax for maximum velocity, right? And if you start adding inhibitors to the mix, the rate of the reaction is going to go down, right? These two different types, competitive and non-competitive inhibitors. A competitive inhibitor is one that binds to the same binding site as the substrate. And so in this little drawing, if this is our enzyme and this is the substrate, a competitive inhibitor would fit in this binding site, right? And so it physically blocks the substrate from gaining access, okay? And if you add more and more of that inhibitor, this line would go down further and further, right? So this graph isn't really showing us how much competitive inhibitor we added, but if we added more, the line would go down further, go down further, go down further, okay? The one catch is, if you add enough substrate, you can eventually get back up to Vmax. Okay, so with just the enzyme by itself, we hit Vmax about here at this concentration. With the inhibitor, we can still hit Vmax, but we have to go further out on the x-axis, add more substrate. The reason is they're competing with each other, right? It's a probability game. The more substrate molecules you have, the more of a probability there is that your substrate will bind to the active site before the inhibitor can. Okay, so competitive inhibitors do slow down the reaction, but they can be outcompeted by adding a lot of substrate. Okay. All right, the non-competitive inhibitor, same concept. It's still slowing down the activity of the enzyme, but the difference is a non-competitive inhibitor binds somewhere other than the active site. Okay, and so the active site would still be open and accessible to the substrate binding, right? Except that when our non-competitive inhibitor binds, the active site changes shape a little bit, right? It's called a conformational change. Sometimes you will see that non-competitive inhibitor called an allosteric inhibitor. The term allosteric just means anything involving a shape change, right? When our allosteric or non-competitive inhibitor binds the enzyme, it creates a ripple effect, right, through the enzyme, and the whole thing sort of twists, right? Imagine like someone getting a little electric shock and their body just sort of contorts a little bit. The whole enzyme is slightly changing its shape, so now the substrate can't bind. Okay? Since they aren't in direct competition with each other, the non-competitive inhibitor gives you a new Vmax, right? That decrease in Vmax is because when this non-competitive or allosteric inhibitor is bound, the enzyme is non-functional. It's out of commission. It doesn't matter how much substrate you add because they're not competing for the same binding site. So it's not that same probability interaction like it was with the competitive inhibitors. Okay. Now, in a biological context, we use inhibitors a handful of different ways. A lot of poisons, like mushroom poisons or snake venoms, are enzyme inhibitors. A lot of medications are enzyme inhibitors. For example, antibiotics all inhibit bacterial enzymes, so it acts like bacteria poison to get rid of the infection. And some inhibitors are also used on purpose, right, inside of cells as a way to regulate how fast enzymes act. Right. So we'll get to some examples of this, but the other thing to remember in the meantime is you can also have activators. Right, so we're seeing that term allosteric again. Remember, it just means shape change. So you can have a molecule that binds and acts like an allosteric inhibitor. Right, we've already seen that. They're saying that this open sort of square conformation in the active site is the active form, and this inactive form is the skinnier rectangle. And so all this diagram is showing you is that if the allosteric inhibitor binds the enzyme, when we say that it stabilizes the inactive form, it really just means that it traps the enzyme in the inactive state. Okay? And if you have an activator is just trapping the enzyme in the active form, right? And so it's encouraging the enzyme to keep a particular shape. Inhibitors favor the inactive shape, and activators favor the active shape. Makes sense. Right, so if cells want to make their chemical reactions go faster or slower, they can use these allosteric inhibitors or activators to control how many enzymes are actually active and how many are turned off. Right. Another important thing to add in, enzymes often require something called coenzymes, and coenzymes are really just organic molecules that bind to the enzyme and are required for its function. So you'll notice vitamins are very, very common coenzymes. If you don't have the vitamin coenzyme to be part of the enzyme complex, it just won't function properly. Right? Another thing to add in, you can also have something called cofactors, which is very similar, but those are usually metal ions, things like magnesium or zinc are common, or iron. So metals are commonly cofactors. Vitamins that are organic molecules are generally coenzymes, but it's the same concept. They're necessary for like an add-on to the enzyme to allow it to function. Okay. All right, last concept, this feedback inhibition. You might also sometimes see it just called negative feedback. This is an example of how inhibitors are used naturally inside of a cell for good reasons. Right? So most people think that inhibitors are a bad thing, but they're often used for good. So in this example, we've got a metabolic pathway where we're starting with some sort of starting material, doesn't really matter what it is, and by the end, we want to produce a product, right? And we've got 4 enzymes along the way that are going to carry out a 4-step reaction. So we start over here with what we can call thing A, and enzyme number 1 converts it into an intermediate that we can call molecule B, and enzyme 2 converts that into molecule C, and enzyme 3 converts that into molecule D, and finally we get our product, which we can call product E. Now, the thing is, product E, we probably want to keep at homeostasis, right? Meaning we want to keep levels of E stable without going too high or too low. What's interesting is the product of the reaction often acts as an inhibitor to one of the enzymes earlier on in the pathway. And so what ends up happening is if you make too much of product E, perfect. It acts as an inhibitor, binds to this enzyme, and that slows down the reaction because we had too much of our product. We want the enzymes to go slower, right? This is very much like, imagine a furnace in a house when it's cold outside, and the product would be heat. What does heat do? If the house gets too hot, thermostat registers heat, and we shut off the furnace. That way you don't make too much, and the house doesn't overheat. Same thing here. This prevents us from making too much of a product in a pathway. Okay? So usually this feedback inhibition works by non-competitive inhibition. So there'll be a binding site on one of these earlier enzymes, and our product goes back and sticks to it, right? And the opposite is true. If we don't have enough product, that just means that we don't have our inhibitor, and the enzymes will go fast, and we'll make more. And what you get in the end is, if we graft the amount of our product, thing E, over time, you always get this sort of hovering pattern, right? Because you go a little bit too high and come back down because the inhibitor kicks in, and then you go a little bit too low and the reaction speeds back up, right? This is how negative feedback or feedback inhibition is used to keep homeostasis of all sorts of products throughout the body. Think ATP and glucose and pH, right? They're all regulated by the use of inhibitors through feedback inhibition. All right, hope this was helpful. As always, keep track of items in the playlist. We're going to move on to another chapter next, and subscribe to the channel to make sure you get all the updates, because there are going to be a lot of videos in this playlist pretty soon.